This chapter will demonstrate how to understand the basics of RNA-seq data collection and processing workflow. Identify the next steps for your particular RNA-seq data. Formulate questions to ask about your RNA-seq data. Bulk RNA-seq data is generated by extracting total RNA and then isolating RNA-specific species by either poly-A-selection, ribodepletion, or size selection. The isolated RNA is then converted to key DNA, so it is more stable for sequencing. This key DNA is used to construct a sequencing library. Lastly, PCR amplification is used to make many copies to use for sequencing. When looking for tools, you will want to see if the algorithms or options available attempt to account for these biases in some way. Poly-A-selection advantages: lower sequencing depth needed, greater exonic coverage. Disadvantages of poly-A-selection is that it does not detect non-poly-A transcripts including murnus, snornus, and sumuncuinous. It obtains less information on immature transcripts. It performs poorly for degraded RNA or formalin-fixed paraffin-embedded epithelial samples biased towards 3' end of transcripts. Cannot be used for prokaryotes. Ribo- advantages are: it is able to detect small and non-polyadenulated RNAs. It detects long and short transcripts, no 3' bias. It has better performance on degraded RNA or FET samples. It is applicable for prokaryotes. It can be applied toward other abundant RNA. The disadvantages of Ribo- is that it will collect more intronic reads and immature RNAs if you are not interested in those, and thus because of the greater quantity of the returned RNA pool, it requires greater sequencing depths. RNA-seq data has various biases introduced to the data upon data generation. RNA targets are more likely to be picked up if they are long, if they are from the 3' end, have a particular GC content, and have a particular read start sequence. When looking for analysis tools, pay attention to what abundance measures the tool expects to be given with respect to what transformations have already been done to your data, if any. In a very general sense, RNA-seq workflows involves first quantification element. You will also need to conduct quality control steps that check the quality of the sequencing done. You may also want to trim and filter out data that is not trustworthy. After you have a set of reliable data, you need to normalize your data. After data has been normalized, you are ready to conduct your downstream analyses. This will be highly dependent on the original goals and questions of your experiment. It may include dimension reduction, differential expression, or any number of other analyses. TOEFIT uses an expectation maximization approach that estimates transcript abundances. Cufflinks is designed to take advantage of p-reads and may use GIF information to identify express transcripts, or can infer transcripts de novo from the mapping data alone. RSEM Express, Sailfish, Callisto, and Salmon quantify expression from transcriptome mapping and allocate multi-mapping reads among transcript and output within sample normalized values corrected for sequencing biases. NERD provides an efficient way of estimating transcript expression from, say, reads with a low memory and computing cost. For DSUC, read count distribution assumes a negative binomial distribution. Raw counts are expected. Replicates are not dealt with. Normalization is done with respect to library size. For Etcher, read count distribution assumes Bayesian methods for negative binomial distribution. Input is raw counts. Yes, can deal with replicates. Normalization is done with respect to library size, tame, roll, upper quartile. For BASIC, read count distribution assumes Bayesian methods for negative binomial distribution. Input is raw counts, and deal with replicates. Library size quantile, for NOISOC, read count distribution is assumed to be non-parametric. Input is raw or normalized counts, does not deal with replicates. Normalization is done with respect to library size, same, TEM, upper quartile.